It's a pleasure to be here. I've been doing this kind of research for quite a long time in terms of looking at components in the environment that have some adverse effects, pollutants primarily, but I've looked at all sorts of toxic constituents, including effects of drugs, but always looking at how these effects may be mediated or protected against using nutrition. So this aspect is something that I'm also been focused on, particularly on the brain level. Many environmental contaminants adversely affect brain function, and particularly younger age groups, but it goes the whole spectrum of all the ages. So in general, we'll talk about, first of all, one important component that is environmental toxicants called Pcbs. I'll give some general information of what these are and their importance and how they enter in the environment, where are they found in the environment, and how long can they remain. And essentially, they remain for a very long time, as you'll be seeing. And we'll focus on a little bit about the health effects in terms of toxicity, particularly at the brain level, but they affect all organs in the body. And something about how to avoid them and how potentially nutrition may prevent the harm, which is one of my areas of research. It's a short talk. It's only about 10-15 minutes, as I've been asked to give, but I'm happy to talk all night on this. So Pcbs are, the acronym is for polychlorinated biphenyls, and this is the structure that you see here. Of these compounds, and they're a whole host. They're not just one chemical compound. They have this kind of ring structure, which makes them quite fat soluble, meaning they associate with fatty components in foods and tissues and in the environment. What makes them foreign to the body and makes them more difficult to metabolize, not only in the human body, but in the environment in general, is they have these chlorine rings. So they have different amounts of chlorine associated with it, which this property makes them more difficult to metabolize in the environment as well as within our own bodies. And that makes them persist for a very long time because these are man-made structures that they're not naturally occurring through nature. That makes them more difficult to break down. There are about 130 what are called congeners, basically referring to that. What makes this even more complex a topic is that there's various numbers of chlorinated substitutions on this ring that makes different compounds, that makes them behave differently both in the body and in the environment, and that adds to the complexity of the toxicity as well. What makes these particularly harmful to the environment and ultimately to human health is that they've been utilized heavily and manufactured since the 1920s. As we'll be talking about, for various purposes. And it's only been banned, so for about 50 years. It was banned in 1979 because of its toxicity that was uncovered. This is a little different from many other toxicants that are more recently been identified and potentially toxic, but because of this long use before they were banned, there's huge accumulation in the environment of these compounds. So there was a Stockholm conference about 10-15 years ago that identified Pcbs as part of what is called the dirty dozen, which are called persistent organic pollutants, or POPs. That refers to a whole set of compounds similar in structure, and they have this chlorinated composition. And these compounds are, a lot of them are pesticides such as DDT, products that are produced also from just combustion. Some of these can be naturally found, such as dioxins, but most of them are produced from industrial processes and have accumulated in the environment and are now banned because of their toxicity and their persistence in the environment. They're also called organochlorines, meaning that organic compounds, they have a carbon associated with them, like normal naturally occurring compounds in nature, but what differentiates them and makes them very difficult to break down are chlorine. There have been subsequently added a whole host of other compounds similar in nature since this conference that was about 10 years ago. So there's what's similar about these is that they all have adverse effects on human health and to the environment as well. So they can adversely affect the health of mammals and birds and wildlife in addition to human health. What makes them difficult is that they persist in the environment because of the chlorine, and also they are stored in fat. So because of their structure, the body responds to, our bodies try to, when there's a fat compound, they try and maintain the compound and absorb it for later on for use, thinking it can be used as for energy purposes. So they are readily stored in fat, and as I said, not readily broken down in nature. So where do they come from? Despite being banned, they've been used and for many, many purposes, and are still used for many purposes. Although from places such as capacitors, transformers, they have a poor chemical reactivity. They're quite stable chemically. They have a very high stability under high temperatures. So they thus, under situations where there's electrical equipment such as transformers, they've been used and are still currently used in that aspect to prevent fires from occurring. But they've been used in a wide variety of products, paints of all sorts, caulking agents, a huge variety of products until they were banned. So where you have problems is when there's waste disposal of electronic equipment or transformers, and if they're poorly maintained, you have runoff that can leach into the soil. You have disposal of consumer products that were not designed for hazardous waste, because in reality, to properly dispose of them, they have to be burnt under very high incinerating conditions, which are specially designed to burn these compounds. So normal breakdowns, such as a fire, for example, will just release more of these products that are broken down from the fire, but equally hazardous. So some of the waste that is in these products that can get into municipal or industrial incinerators that contain PCBs, if not properly designed to break them down, will just aggravate the problem. So as I mentioned, they don't break down, and they recycle between air, water, and soil, and there's a constant movement of these PCBs into the various areas, and they can vaporize to a small degree, and this allows them to be carried in long distances and being released. And what tragically you have is, particularly in the northern regions, such as the Arctic, quite a large release of these compounds because it gets into the air and wind currents. And then, because of the cold temperatures, gets deposited into the water. Under warmer climates, the PCBs will evaporate from the water and then recirculate in that manner, but not in the colder climates where you have they will not readily vaporize. And so there's a concentration of these compounds in the Arctic for that reason. So there are some machinery that are still in use: fluorescent light fixtures, electrical appliances, transformers, as I said, that they still are used. And it's estimated that there's approximately about 130 tons presently worldwide, a million tons, 130 million tons of PCBs, of which approximately 70% are either in storage or in current use in equipment. So there's still a huge environmental burden of these compounds. So as I said, they readily dissolve in fat compounds, and so this allows organisms to accumulate them. They have very slow breakdown because there's no natural enzymatic metabolic machinery to be able to break them down. And they are recognized as fats, which our bodies naturally try to conserve for survival. And there's no differentiation of our bodies from compounds that are fat soluble, such as vitamins A and E, versus toxic compounds, such as Pcbs. So they increase as you go up in the food chain. The ability to concentrate goes up, then the smaller animal is eaten by the larger animal, and so on and so forth. And as you go up the food chain, the concentrations can accumulate. So, so the water concentrations can be relatively much lower versus what is in the aquatic concentrations of bioorganisms. So up to 2,000 to 1,000,000 times higher concentrations of Pcbs are seen in organisms in the water versus the water concentration itself. Where we are exposed, well, a large component is the food because of the fat soluble concentrations. Fatty foods, meats, dairy products, fats and oils will accumulate these to a greater extent. You can have, depending on the workplace and how old the building is, you can have significant accumulation of these Pcbs in indoor air, particularly in the dust. This is where, in fact, younger children or toddlers might be exposed to higher concentrations as they're walking on or being on the ground level or crawling, and they can have higher concentrations, particularly in older buildings where you have still paints and caulking agents and electrical equipment that is older that can leak out. And this can also happen in industrial context or school contexts. Occupational context where you have older buildings. In some places in the US, some of these buildings are actually banned from being entered into because of the high Pcb concentrations. Where there is exposure, unfortunately, is also in pregnancy and lactation because of the fact that there's mobilization of fat stores during those periods of life. So in lactation, for example, the maternal fat stores are mobilized to release fat into the maternal milk. Milk is a very high fat source, which is important for the feeding infant. However, this also releases from the fat stores the Pcbs. And people who go through rapid weight loss, for example, can have significantly high concentrations of Pcbs in their bloodstreams for a period of time due to the release of them through the rapid fat loss in the tissues. However, the studies to date say despite that, there are more benefits than harm for the lactating infant. Depending on if there was a woman who was occupationally exposed or to very high concentrations, there can be some adverse consequences in that manner, potentially to the offspring. And that's an area that's still under investigation, particularly in, as I'll be talking about, in certain situations, occupationally or dietarily, that the mother may be exposed to. So there is, the good news is certainly Health Canada, as many other governmental organizations in other countries, they do have carry out what are called Canadian, in this context, total diet studies. In Canada this happens, and you have other countries in the world that this is monitoring of contaminants in the food supply. And so that essentially what happens in this context is they take from different areas of the country the food supply, they prepare the foods, of a wide variety of foods, prepared and made composites according to the meals people take in, and then they actually measure for various toxicants within the food to ensure that the food supply is safe. Typically, under this context, the average intake is only about half a microgram. That's 1/1,000,000th of a gram. So the concentrations generally are thought to be deemed in a safe area in the Canadian context. And the other good news is that the concentrations have declined since them being banned about 25 years ago. The concentrations both in food and the amount taken in through as measured by the total dietary studies, as well as the human tissue levels. And so the Health Canada has determined that the concentrations generally are safe, and there's no fish is where you potentially have higher concentrations because of the Pcbs in the water supply. Generally, fish will provide maybe 40 to 50% of the concentrations that people are normally exposed to. Unless they don't eat much fish, then they won't have much lower contribution from that source. However, there is one particular segment of the population that are at higher risk. Those who eat lots of sports fish that's problematic, or wildlife, or marine mammals, such as, unfortunately, the Inuit, for example, in the Arctic regions. There may be significantly higher levels of PCBs exposures. And in fact, Health Canada does have recommendations of how much sport fish should be eaten according to the water supply and the concentrations of PCBs and other toxicants in that water supply. And so there are advisories for specific lakes and regions and of types of fish they can be eaten or avoided and how much they should eat. Generally, certainly for the pregnant lactating situation or for young children, sports fish, fish eaten from recreation fishermen in these waters generally is contraindicated completely because of the relatively high levels of PCBs that may occur. This is particularly problematic for children, but it could be even a problem if women during adolescence, for instance, are eating a lot of sports fish. That burden is actually could be quite significant because of the fat accumulation in their tissues. Once they start to have, during the pregnancy and lactation phase, that's where the release of the PCBs occurs. So in fact, preventing women from taking in earlier years of PCBs through that mechanism is important. Also, it's in the Inuit and areas where there's subsistence living on foods that are traditional foods that is potentially problematic from the larger marine mammals, pilot whales, polar bears, these larger fish that they will accumulate significant amounts of PCBs, and there are now advisories for intakes to be lower in those regions for those type of marine mammals or fish. There are still ongoing investigations because of the fact that there are gradations of exposure, not only through diet, but through runoff in certain areas that may get into the food or water supply or in the air in certain regions, that these relatively low exposures have started to show some relationships with adverse outcomes later on in life, such as particularly in the newborn and young children, showing problems in terms of learning, facial recognition problems, memory, impaired psychomotor skills. These have been linked in more recent years with higher exposures of these children either during lactation or during pregnancy, and concentrations have been related to these adverse outcomes. So it's still a complex issue because there are complexities in terms of what the exposures there are. These breakdown products and these PCBs are actually highly complex and difficult to measure in the body itself, and their risks are still being uncovered. There are a wide range of toxicities. They're stated to be probable carcinogens, that is cancer-causing substances in humans. There's been a number of acute effects. There's inflammatory acne that has been seen from occupational exposures or from during fires due to the absorption. There have been some spills of PCBs into oils in certain populations, and that causes major developmental problems in the offspring of women who are exposed to the spills. And even 10, 15 years later, these women or men that were exposed to these spills still have several-fold higher concentrations in their body from that exposure because of the poor breakdown of these products. They're problematic also because they, one of the mechanisms that they adversely affect is the fact that they mimic or block a number of natural occurring hormones. They mimic the effects of estrogen, thyroid hormones, and in that manner actually can have quite severe effects on immune system functioning, on growth and development, and in terms of brain function as well. That's been related with higher concentrations. Chronic exposure to greater than normal has been seen in terms of in males, sperm integrity, sperm count. Recent publications have shown that women who have trouble conceiving have been exposed with higher amounts of Pcbs, including having problems with in vitro fertilization of the eggs due to exposure of Pcbs. Immune system problems have been noted with these compounds, so increased respiratory and ear infections in children, and recent publication shows that the ability of children to respond to diphtheria and other immunizations, the amount of antibodies that they produce to these immunizations is lower than it should be, and that the children may not be perfectly protected as other children due to the exposure of Pcbs. And an area that I look at is looking at brain function as affected by Pcbs, because there's good evidence that higher exposure can have all these adverse effects on development. Some of the studies, for example, in the Great Lakes region is where you had a large accumulation, deposition of Pcbs over a long period of time. And when women have been taking in sports fish through sports fishery, then this accumulates in the placental tissue, and in the boys exposed during pregnancy to these higher levels of Pcbs have, depending on the dose, sequentially adversely affected intellectual functioning. This has not only been seen in children, but there's indications that the presence of these compounds in higher concentrations in adults, including older adults and elderly individuals, has been linked with neurodegenerative disorders. So some of the recent publications are Pcbs as risk factors for attention deficit disorder and hyperactivity. There's been a consistent link with Parkinson's disease and exposure to these compounds. There's also evidence of neuropsychological effects in terms of well-being, hyperactivity being elevated with higher blood levels of Pcbs as well. So what's the way to avoid these compounds? As I say, they tend to be stored in animal fat, so lean fat foods, and lean meats, poultry, dairy products that have lower fat content. Legumes, nuts, and seeds are a good alternative source of fat. There is a suggestion that not to avoid fish altogether, because there's it's a double-edged sword in that avoidance of fish could be counterproductive because of the important nutritional properties of seafood, including improvement of brain function due to the type of fatty acids that are present in fish, such as they're termed omega-3 fatty acids that are important for optimal brain function. On the other hand, the smaller fish will not accumulate significant amounts of Pcbs to have toxicological outcomes. So the concentrations are relatively low, such as sardines as an example, or mackerel. These types of fish has relatively small concentrations that are toxicologically insignificant, unless they've been from a source that is contaminated, such as in sports fishery in lake regions. So that's the idea is to limit families that should be aware of advisories if they catch fish locally, what the risks are. Because they are found in the fat, removal of the fat, and also using baking or broiling or microwave cookery will remove a lot of the PCBs from the release of fat. On the other hand, if you fry this in fat and eat the fat, then you'll just not be able to remove and continue to eat the PCBs. So avoiding the fat content will avoid a lot of the problems in terms of PCBs in fish as well. So children can be exposed. There's indications that if they start to eat a little bit of soil, which children sometimes do, or clay, and then it's in a contaminated area, this could be a risk for them. It's been noted that if they're occupationally, just dust from exposure of individuals who work occupationally, this can contribute to increased accumulation of PCBs in house dust, or if there are older homes that have accumulated this through the paint or the caulking or other sources in the homes. Older homes are more at risk in that regard. Unfortunately, if you avoid all PCBs through pregnancy, it will have no impact whatsoever. Even if you took it out of your diet supply 100%, you would still have equivalent amounts of PCBs during pregnancy and lactation, because it's not the amount taken in during pregnancy and lactation is irrelevant. It's the lifetime exposure that is stored in the fat that makes the impact. So making a change to dietarily avoid them will not really make any impact in terms of that during pregnancy or lactation. And in fact, the studies have indicated that women who take in a lot of sports fish during adolescent period will accumulate significant amounts of PCBs, which then could be unfortunately released and lead to higher concentrations in the blood during the pregnancy and lactation context, and expose the infants in that way. So we're looking at PCBs in terms of what can you do dietarily to help counteract some of these exposures. We look at one aspect. There's a variety of mechanisms by which they act, including what is called free radicals. Our bodies actually metabolize a lot of toxic foreign compounds, and during that metabolism, it could be actually counterproductive to our bodies, producing what are called free radicals. These are molecules that are highly reactive, can damage our proteins, DNA, and fat, and cause damage to tissues such as the brain. So we've been looking at antioxidants that are found, particularly plant-based antioxidants that are found high and rich in certain plant products, and to see whether this could protect against some of the toxicities associated with PCBs, particularly at the brain level. One problem with it's a very complex issue, because the metabolism generates all sorts of products from PCBs that react with a number of hormone receptors, and we still have a problem identifying some of these products because they're very complex to measure within the body, and this is an area that is likely to be one aspect that leads to the toxicity is the metabolism of these. So sometimes it's not the PCB itself, it's its metabolism, and it's the metabolites that are broken and released from the PCBs that could be quite toxic. So we've published looking at neurochemical imbalances showing the antioxidants in the form of the essential mineral selenium and vitamin E in an animal model protected some against the neurochemical imbalances, not only of PCBs, but another toxicant associated with seafood, methylmercury. Others have shown that higher omega-3 fats found from plant sources protect against PCB damage, that PCBs have been linked with increased risk of obesity and hypercholesteremia, high blood cholesterol, as well as diabetes due to their impact on hormonal receptors. And omega-3 fats found in fish, per se, it can be protective as long as you eat the right fish. So you don't eat large fish such as tilefish or shark. It's the smaller fish because they actually don't have much omega-3 fatty acids in the first place. They tend to accumulate actually more of the toxicants. Swordfish is another example of a large fish that doesn't have much in omega-3 fatty acids but is rich in PCBs. So those kinds of fish you don't want to readily eat regularly, as opposed to the smaller type of fish like sardines. And there's one compound we look at, which is called quercetin. There's these so-called phytochemicals. These are chemicals found in plant products that are very protective on many levels for the body, and there's been recent publication that this might help against neurodegeneration is what we're looking at. What we do is, in collaboration with Satya Prakash in biomedical engineering, is we have a artificial human gut system that we study how PCBs are metabolized and how nutrients may protect against PCBs. So essentially you have a computer-controlled system where food comes in, it goes through the stomach, small intestine. Of particular interest for us is the large intestine, where there are a lot of microbes being present. And we look at the fact that a large part of metabolism that is actually central for health happens at the colon level due to the presence of bacteria. Those bacteria are exceedingly important in terms of protection against disease, but also are an organ in a sense that they actually metabolize toxicants. And they may lead to, by changing the bacterial flora, with what we're looking at dietarily, you may actually change the metabolism of PCBs so they're either less well absorbed or less toxic when they enter the body. So we look at this, and actually we have a model system that simulates what happens through the whole metabolism of the human gut and post-absorption. So the PCBs that or polyphenols are a compound that are found in plant products that we're looking at that might be protective for brain health. These products are actually taken in from the artificial gut that I've just shown. The metabolism of these PCBs may be altered by the bacteria to actually be more toxic. Or conversely, your bacterial flora to be healthy for your body has to be maintain a certain proportion of types of bacteria. PCBs, one way they may be causing harm in terms of risk of neurochemical imbalances in the brain or causing problems with high blood cholesterol or obesity, might be mediated by the fact that they can be quite toxic to your intestinal flora. In fact, this has been seen with soil microorganisms exposed to PCBs, that they are adversely affected. We think that same thing may be happening in the gut. So we're looking at compounds such as polyphenols, which are found in nice places in rich concentrations like coffee and red wine and dark chocolate. All your favorite foods have lots of polyphenols, including fruits and vegetables. And these compounds may be protecting potentially the bacteria. In addition, there's a major problem of testing for PCBs in terms of animal models are not particularly necessarily effective because they have different kinds of bacteria in their guts, they have different types of metabolism and sensitivity of neural tissues. So we use human tissues to look at what the absorption is. You can see the absorption of PCBs or polyphenols as protective agents. And then after you've absorbed something, it goes through liver metabolism. So we have human liver metabolism analogy that these compounds go through. And then we have a three-dimensional brain structures that are human brain structures that have all the same cell populations that you see in the brain. And then we look at how PCBs might be affecting the outgrowths of these brain cells. These outgrowths really are neuron cells in response to learning and memory start to have these long elongations called dendrites, and you can measure for this, and the ability to produce these dendrites is a symptom of health, whereas PCBs are known to inhibit these. So we can look at the protective effects of polyphenols and other nutritional compounds to allow these dendrites to develop more properly. Inhibition of these dendrites will lead to these outcomes that I've talked about, problems with learning and memory. And these are cells that actually we have that are very different. You may have thought that all our brain cells, once established, don't replicate, but in reality, there are cells called progenitor cells that are like embryonic neurons that are completely developed all through your life. And for optimal health, these are maintained, and you can maintain learning and memory to an optimal fashion as long as these progenitor cells are maintained to be healthy. PCBs will adversely affect these progenitor cells, these embryonic neuron cells, and that will inhibit the learning process. So the ideal is to protect against this, potentially through nutrition, so that these learning and these progenitor cells will maintain themselves throughout life. So in conclusion, they have declined since the 1970s, but it's increasingly clear that the concentrations in the environment and tissues still are producing harm and are related to all sorts of adverse outcomes, including more recently metabolic disorders such as diabetes, and high blood cholesterol has been linked in addition to effects on the brain. It's clear that exposure during pregnancy and lactation from the PCBs at higher concentrations does have adverse effects on learning. And we're looking at how nutrition can potentially protect against this risk, since we may be exposed, or women may be exposed to these compounds inadvertently, and there may be a dietary regimens or nutritional strategies to decrease the risk. So thank you for your attention.